So it's really an honor, and it's true. I was completely surprised when I got this. It's an honor to be here in Montreal for ISMB, and I really love this country and I love this city. So to the Canadians here, I'm so pleased to be here. It's true, people have a big impact on our lives, and one of the things that I want to do in this talk is give you some presence at the end with some open problems. So, it's not just a talk talking about what I've done. I want you to go away with perhaps some ideas for your own research or for your students. And again, thank you to the ISCB for the award. Thank you. So phylogenomics is phylogeny estimation at the genome scale, and it creates all sorts of interesting challenges. I've been on several different phylogenomics projects. I just want to give you an example of one, the avian phylogenomics project, which had roughly 48 birds, whole genomes. So with only 48 birds, you would think it's not a big data set. But trying to do a phylogeny at this scale turned out to be really computationally challenging. And even without taking any heterogeneity into an account, from a computational perspective, the maximum likelihood tree on that data set took 250 CPU years. So it's a computationally challenging problem as well as a statistically challenging problem. And I'm going to be emphasizing both ideas and challenges in this talk. So large data sets are difficult for phylogeny estimation in multiple ways. One is that there are two dimensions of large, the number of loci, or you can think of it as the number of sites in a long alignment, but also the number of individuals in your tree. So the number of leaves in the tree you're going to be computing. Either one can be large and make the problem challenging. And when they're both large, it's extremely difficult. You can have missing data, you can have heterogeneity across the tree, across the sites, and you can have statistical approaches like maximum likelihood and Bayesian estimation, which really make for computationally challenging approaches. So this talk is really about divide and conquer. So you can think of it as a computer science technique. Can divide and conquer help ameliorate these challenges? And they actually can, but it's non-trivial. It's not a straightforward just do divide and conquer and now everything's easy. And I'm going to try to give you some ideas about what has worked, what hasn't quite worked, and where there's opportunity to improve things. So I'm going to start with divide and conquer using supertrees. So supertrees are a basic approach where the input is a set of overlapping trees, so that is trees that have common leaf sets. They don't have to have all the leaves, but they have some leaves in common. And you're trying to put them into a tree on the entire data set. So in a divide and conquer strategy, you have a large data set, you divide into subsets, you can get trees on the subsets, and then you can combine them with a supertree method. That's what this topic is going to be about. And I'm starting with this approach because it's been traditionally well understood in the computational biology world, and there's lots of techniques out there. But then I'm going to show you that there are limitations, and so we're going to go to a new technique for doing divide and conquer. Okay, so a cartoon of sequence evolution is you have a sequence at the root of the tree, and it evolves down the tree with substitutions. And so when you evolve down the tree with substitutions, you end up with leaves at the modern day time from which you're trying to reconstruct the tree. So this is a cartoon. There's no insertions, there's no deletions, there's no rearrangements, there's nothing particularly interesting happening. But can you reconstruct the tree from the sequences at the leaves? Even without any special things happening, you would think this might be possible or might be very difficult. So, whoops, how do I get rid of that? How do you get from the sequences at the leaves to the tree? And I want you to notice that this tree is not rooted. The tree that generates the data and you're trying to reconstruct is rooted, but you're going to actually be satisfied with getting the unrooted tree because of time reversibility in most sequence evolution models. So you'll consider yourself correct if the unrooted tree is exactly correct. To discuss this as a statistical problem, you need a model of evolution. There's a very standard model called Jukes-Kanner, which is for DNA. And basically, in the simplest form, there's a random nucleotide at the root. All of the sites evolve under the same process. As a site evolves down the tree, it changes on an edge with a probability for that edge. If it changes to one of the other nucleotides with equal probability. Now that's very simple, and you can make it more realistic by having substitution rate matrices. You can actually make it additionally realistic by allowing rate variation across sites. But basically, this is the underlying type of model that everyone uses. And under this model, you can ask, can you reconstruct the tree? So as a statistical problem, you want to reconstruct the unrooted model tree from the sequences you see at the leaves, given that you know something about the model, but you don't know the parameters of the model. You don't know the topology, you don't know the rates. So there's many different methods. There's maximum likelihood, which is to try to find the model tree that has the highest probability of generating the data. There's Bayesian methods, which are based upon the same model, but are doing a random walk to sample from the distribution. There's distance-based methods, such as neighbor joining, that compute a distance from the sequences and then compute a tree. And then there's maximum parsimony, which tries to find the tree with the minimum number of changes. Almost everything you want to do in phylogeny is NP-hard, almost everything. Distance-based methods in some cases are not NP-hard, but most of the optimization problems are NP-hard, and so most of the time you're dealing with heuristics. So when you talk about a method, the properties you are interested in understanding are statistical properties, such as statistical consistency, its sample complexity, and I'm going to illustrate these two concepts in a minute. There's computational performance, so runtime and memory usage, and then there's accuracy. So what do we mean by accuracy? We want to look at what happens on data. And as Aida was saying, I really discovered this difference between theory and data evaluation with Ken Rice, who was a postdoc of mine. Amazing differences between theory and what you see on data. So data, you can have simulated data where you know the truth because you simulate the evolution, and you can have biological data where you don't know the truth. And it's challenging. Both of them have some advantages, but neither has only advantages. So you need to do both. So let's talk about what happens if you do a simulation. You have a model tree, you evolve sequences on the model tree, you construct a tree. And now that constructed tree is unrooted, the true tree is rooted. You compare the two trees and you mark the edges as either being correct or incorrect. In the inferred tree, I've drawn an FP, which means false positive, on the edge that splits S3 and S5 from S1, 2, and 4. And that's because that split does not appear in the true tree. Okay, so that's a false positive. The other edge in the inferred tree does exist in the true tree, and so that's a true positive. So you're looking at the edges and saying which edges are correct and which edges are not correct. And you can talk about the error rate, the false positive error rate, the false negative error rate, and when the trees are both fully resolved, then those are the same rate. So when I show you error rates in the future, I'm going to be talking about this kind of error rate. So statistical consistency under a model is a theoretical statement that requires a proof, and it's a statement that the parameter you're trying to estimate will have error that goes to zero, provably, as the data goes to infinity. So if you're trying to estimate the tree topology, you want the topological difference error rate to go to zero as the amount of data you have goes to infinity. Now, for sequences in an alignment, the amount of data is the length of the alignment. So as the length of the alignment goes, you want the error to go to zero. You can't demonstrate it in a simulation. You need a theoretical proof. It turns out that for most cases, proving statistical consistency is actually pretty easy. So I just want you to realize statistical consistency is like maybe a requirement, but it actually is not hard to establish for many methods. But you could also debate whether or not it's a requirement. Anyway, this is a very, very basic property. So sample complexity is actually getting at how much data you need to get that tree with high probability. or rather how much data suffices. You can look at it in either way. So if you remember that in Jukes-Kantor, every edge has a probability of change that is going to be between a value f and a value g. So the smallest probability of change would be f, the largest probability of change would be g. And if f is very small, you're going to need very long sequences. If g is very big, you're going to need very long sequences. But once you fix f and g, you can sort of talk about the sample complexity only now as a function of the number of leaves. So you can say, is that sample complexity polynomial? Is it exponential? You can get an idea about how much data you need. So that's another perspective, how quickly you are approaching zero. So absolute fast converging methods is a term that we developed that says, even if you don't know f and g, your algorithm has this guarantee that not only is it statistically consistent, but it will recover the tree with probability at least 1 minus epsilon from a polynomial number of sites. Polynomial in n once you fix f and g, okay? So it's not runtime, it's sample complexity. So sample complexity could be exponential, it could be polynomial, it could be something else. So we're interested in methods that have good sample complexity because if they have good sample complexity, maybe they also have good accuracy on data. It gets a theoretical perspective. Can I get some help here?
I'm just going to close everything else so that it's. We're going to close everything so that there's nothing else that can be opened to pop up like that again. That one.
Thank you.
You're good.
Okay, so look what happened.
If it pops up, you can just click on the screen and it will keep going.
Okay, I hope that's not going to happen. I learned the trick. Okay, good. Okay, sample complexity. Again, we're talking about how do we evaluate methods. We want runtime to not be too bad. We want sample complexity to be not very large. We want accuracy on data. Right now, I'm just talking about theory. So distance-based estimates are actually the easiest type of method to evaluate. You have a data set of sequences, you compute distances between the sequences, and you compute those distances under the model so that it will converge as the sequence length goes to infinity to what's called an additive matrix, one that actually realizes the tree. And then from your estimate of that distance matrix, you can compute the tree. And it's really easy to prove that methods like neighbor joining are statistically consistent. Oh, it happened again. Okay, it's really easy to prove that methods based upon distances are statistically consistent. It's also not too hard to evaluate their sample complexity. So now I'm going to be telling you about work that was done, quite a while ago. Peter Erdős, not Paul, but Peter Erdős, Laszlo Székely, Mike Steele, and I proved, along with Kevin Addison, some guarantees about neighbor joining being correct with probability at least 1 minus epsilon, given sequence lengths that are exponential in the largest leaf-to-leaf distance in the tree. So that's great. You have a proof of statistical consistency, but that upper bound on the sequence length that suffice for accuracy is a little bit disturbing because the exponential is large. Then there was actually a lower bound established by Lacey-Knowles that showed also exponential. So you have this situation where you have a method that's statistically consistent, but it has this vulnerability to large distances. So in a simulation study that I did with my former student, Loai Nakla, who's now Dean of Engineering at Rice and also an ISCB fellow, we showed on data that as the diameter increases, and this is by having a fixed range of f and g, and you just take larger and larger trees, as the diameter increases, the error rate goes up for neighbor joining. So neighbor joining is a very basic method, but it has this problem of accuracy decreasing with high diameter. So a bunch of methods were developed, again, starting in 1997, but lots of methods over the years were developed that are provably absolute fast converging. So with that development over the time, we have this nice understanding of the ability to make methods that have this absolute fast converging property, which means they should do better on data. But I'm going to describe one divide and conquer technique that uses a supertree method. So the idea is you have a data set, you divide into subsets, you get trees on the subsets, and you combine them using a supertree method. The algorithm that you are using matters for the combination of the trees, but how you divide into subsets matters. And what this particular technique does is even more complex. It decides on the distance it's willing to consider as the maximum distance. It decomposes based upon that distance. It gets trees on subsets that have that maximum diameter, combines them, and then puts that into a box. And then it changes that maximum distance to a larger value, then a larger value, then a larger value. You get a collection of trees, one for each possible diameter. And from that collection of trees, you then pick one tree, the tree that has the best score. This is a somewhat complicated scenario, but what's really nice is that you now have a guarantee. The guarantee is that if your base method is statistically consistent, so is this pipeline, the DCM boosted version. If your base method is exponentially converging, the DCM version is absolute fast converging. It's a way of changing a method from being exponentially converging to absolute fast converging. And that's the theorem. If it's statistically consistent, it maintains it. If it's exponentially converging, it's now absolute fast converging. And neighbor joining as the base method in this framework now becomes absolute fast converging. Back to data, this is what we had for neighbor joining. This is what we have for DCM boosted neighbor joining. So we have this nice blending of theory and data supporting that understanding. Okay, so are we done? Have we come up with a solution that now can be used on any scenario? And the answer is unfortunately no. And really the reason is based upon what we see on data. If you look at maximum likelihood, it's an NP-hard optimization problem. We have good heuristics, but even not perfect heuristics that are not guaranteed to solve the problem are better on data than this DCM neighbor joining. So from a theoretical perspective, we have an improvement. We have polynomial time, we've got nice theory, it should be fine. But in practice on data, maximum likelihood is simply better. So if you keep comparing to neighbor joining, you think you've solved something. If you now compare to maximum likelihood, you realize you haven't finished the problem. So it's polynomial time, it scales to large data sets, but it's not as good. So are there other divide and conquer approaches that we could use instead of this DCM boosting that would help maximum likelihood? Because maximum likelihood is computationally challenging. And now we get down to what's known in supertree approaches. The most well-known ones are things like Robinson-Fould supertrees, matrix representation with parsimony, matrix representation with likelihood. These are all NP-hard optimization problems. And if you use them for the supertree step, you get computationally extremely expensive, do not scale. So basically, supertree methods people have given up on using supertree methods, I think, for really large-scale phylogeny, okay? Supertree methods reached their limit. We need better supertree methods. The current ones just aren't sufficient. Which brings me to part two. So part two is very new. Most likely, maybe only one or two people in the audience know about what I'm going to tell you about right now. I know two people who do. Maybe there could be a third. But that's it. It's very new stuff. So DTM stands for disjoint tree merger. You have to remember that with supertree methods, the input trees share leaf sets. With disjoint tree mergers, they will not share any leaf sets. So this whole approach was developed by Erin Malloy, a former student of mine, now at the University of Maryland. And the idea is you have a data set, and you divide into disjoint sets. Now, on those disjoint sets, you can construct trees. When you construct trees on the disjoint sets, they don't share any leaves. So if you want to combine them, you cannot use the supertree method. The supertree methods inherently assume that the leaves have some overlap. Otherwise, how do you figure out how to combine them? So what you need instead is some kind of auxiliary information. So when Erin did the study, she used distance matrices. So you can take your input, whatever it is, and compute a distance matrix on your leaves. But then instead of running neighbor joining, after you construct trees on the subsets, which those trees can be based upon maximum likelihood, they can be based upon anything you like. They can be much more expensive on the subsets because they're smaller. And now when you combine them, you're able to use something that's fast to compute with something that's expensive to compute, but the expensive part's on small things. And now you're using the auxiliary information to guide how you combine the trees. Now, her first technique was based upon just modifying neighbor joining, and then she did something that modified that. And then other people have done other methods as well. But I'm going to take you through the basic ideas. So the first understanding you have to have is that you can have by the way, the subset trees cannot change. When you merge them, you're not allowed to change them. So all you're allowed to do is put them together in some way or another. And I want you to see that these trees, Ti, Tj, and Tk, when they're merged, they can be merged in more than one way. In the top merged tree, they're being merged by just adding edges between the trees. So the red leaves stay together, the blue leaves stay together, and then the black leaves stay together. But in the bottom tree, they're blended, okay? If you look at the red leaves, you still get A, B versus C, D, but you have blue leaves interspersed. So this kind of blending changes what's possible. And it's really important to understand the potential that blending gives you. So if every tree is correct, if every subtree is correct, there will be a blended version that matches the true tree. There may not be an unblended version that matches the true tree. So she has a nice theorem that if you do both steps correctly and you use one of her methods, the NJ merge or tree merge, then you have a statistically consistent pipeline. So if the subtrees are computed using statistically consistent methods, if the auxiliary information is computed using statistically consistent methods, then the combination is statistically consistent. Her methods do not allow full blending. Important to realize. Even so, she has a guarantee. Over the years, it started again with Erin, with NJ merge, and then with tree merge. Then there was a method called constrained ink that was developed with Satish Rao and students of his and mine. And then we developed more recently, Vlad Smirnov, a former student of mine, developed something called the guide tree merger. What's really important to realize is the guide tree merger does not allow any blending at all. It only adds edges between the subtrees. The only method that allows full blending is the constrained ink method. NJ merge and tree merge are in between. So why am I emphasizing this? When Vlad came to me and said he had an idea for this disjoint tree merger and that it would not allow any blending, would just add edges, I said, this can't work. I mean, it just will not give you good results. And I'm going to show you that I was wrong. So it's sometimes really surprising, but your students can come up with ideas that just you don't believe that they can work, and they can be so much better than anything you can come up with. So again, the students can bring you wonderful presents. Okay, the guide tree merger. The input is a set of trees on disjoint leaf sets, and you have a tree on the full data set, and you want to merge these trees to make them as close as possible to that guide tree. If you allow blending, this is an NP-hard problem. What Vlad showed is that if you don't allow blending, it's polynomial. So you go from NP-hard to polynomial by just not allowing blending. And what's really way more important is that if you have your guide tree that's statistically consistently estimated, you still have a statistically consistent method, and you actually have great performance in practice. Very strange. So I'm going to now give an example of how this works in the species tree context. So in the species tree context, it's not like Jukes canner evolution. You have to deal with discord across the genome. Different parts of the genome have different trees. This is very well established in biology. It happens for different reasons. One of them is incomplete lineage sorting, which is basically when you have a population rather than a single individual, okay? Different members of the population will have different properties, and their trees will actually coalesce within the species tree at different points in time. So you're just going to have these different trees. There's also gene duplication and loss, this horizontal gene transfer. All of these make for different trees across the genome. I'm just going to talk a little bit about the ILS case, incomplete lineage sorting, because for incomplete lineage sorting, we actually have very good theory. We have statistically consistent methods, and it's so endemic that we need to address it. So this thing called the multi-species coalescent, which models incomplete lineage sorting, plus GTR, the generalized time reversible model for single site evolution. So basically you think a species tree creates gene trees. The gene trees evolve sequences, and what you get to look at is at the bottom of this process, just the sequence alignments, one alignment per gene. And from that collection of alignments, you want to reconstruct the tree. So again, this is a statistical problem now. And you can see this is an example of a species tree in which the gene tree, which is outlined in different colors, is actually different from the species tree. Now, the traditional approach, and you remember I showed this thing of the avian phylogeny, and I said the maximum likelihood tree took 250 CPU years? That was based upon this traditional approach, that you take the different sequences for the different genes, you align them, you concatenate them, and then you run a maximum likelihood method on that, okay? So the problem with this concatenation approach is that it can be statistically inconsistent, and even worse, it can be positively misleading. It can converge to the wrong tree as data goes to infinity. That is disturbing. On the other hand, in practice, it's not often as bad as the theories suggest it might be. So concatenation is still very much in vogue. But Astral is a method that a former student of mine, Siavash Mirarov, developed, which is provably statistically consistent for computing the species tree under the multi-species coalescent. And it's basically, you take all the gene trees that you've estimated, one for each gene. Gene trees, by the way, just means a tree on a given locus. And you then try to find the tree that has the most quartet trees in common. Quartet trees are just four-leaf trees from the gene trees. That problem is NP hard, but if you constrain the search space to say that the species tree should take its bipartitions from the gene trees, you have a polynomial time problem, and it's now statistically consistent. So you have a statistically consistent polynomial time algorithm, and it's become the method in practice across the world for estimating species trees. And it's gone through tremendous development. And by the way, Siavash is here, so if you want to find him and ask him questions, you can. So now this concatenation approach is one major approach. The other one is really Astral. And what Vlad Smirnov did is study the application of this divide and conquer strategy using his guide tree merger with Astral. So Astral is going to be used to construct species trees on subsets. And then the guide tree is going to be a distance-based tree from NJST. And we're going to combine the Astral subset trees together using that guide tree. Okay, so that's the idea. And here's the set of results that we published for this combination. So on the left side, you see a bar graph. The y-axis is the error rate, so it's how much of the tree is missing. You have three different subsets, which depend upon how many genes you're looking at. So it's 10, 25, and 1,000, okay? So as the number of genes increases, all the methods become more accurate. The green bar is this pipeline that takes the NJST tree as the guide tree, gets Astral trees on the subsets, and then combines them. The dark blue is Astral, which is Astral run by itself without this kind of pipeline, then Astrid and NGST are two distance-based methods that are very similar. And what you can see is that on 10 genes, this pipeline is more accurate than the other methods by far, even though it's based upon the other methods. So you're somehow getting more accuracy by doing a divide and conquer pipeline, which is very interesting. It doesn't usually happen that things become more accurate. As you increase the number of genes, you still remain more accurate, but the gap changes. So what we're seeing is a method that is fast, that is statistically consistent. So it's polynomial time, it's statistically consistent, and it's actually more accurate because of the divide and conquer. I have a hypothesis why this is happening, but I'll put that at the end. So we also looked at the runtime comparison, and the runtime comparison is huge. For example, at the 1,000 gene case, if you do Astral by itself, it was 153,000 seconds. But if you do this pipeline, it's 8,000 seconds. So there's this big drop in runtime, and there's an improvement in accuracy. Interesting. So what about scaling maximum likelihood? Let's go back to the first thing we were looking at. Can we get a better maximum likelihood method? What if we looked at, on the subsets, instead of running Astral, we ran RAxML or IQtree or some other maximum likelihood code? So a good maximum likelihood method on the subsets and a faster one for the auxiliary information. So the fastest maximum likelihood heuristic is FastTree, or it's called FastTree 2. But IQtree is also sometimes reasonably fast. RAxML is the one that's really slow. So what happens there? Where do we get an advantage? And this is a study that we published looking, the top is, this is all error rate, and it's all simulated data. So I'm just going to tell you about the different simulated data sets. 1000 M1HF is 1,000 sequences with a high rate of evolution, but fragmentary sequences in part. So some fraction of the sequences are short, the others are full length. The RNA-Sim 1000 and RNA-Sim 10,000 and RNA-Sim 50,000, they're all from this complex simulating protocol that Junyoung Kim at UPenn developed, which is actually not under an IID model. It has, there's dependencies, there's selection going on. It's actually a model that's designed to replicate RNA evolution to maintain the structure. So it's much more complex. Then the COX-1 HET is heterotachy. So we have these different evolutionary models, all of them with some challenge. So in red, I'm showing FastTree, which is the only fast method really. IQtree is in orange, RAxML is in blue, and this pipeline is GTM. The pipeline is going to begin with FastTree if it's a small data set. It's going to begin with IQtree if it's a small data set. It's going to begin with FastTree if it's a large data set, and it's going to run IQtree on the subsets. So on the 1000 M1, FastTree is very high error. This is not good. The red bar is very high error. So that's what you see over here. The next worst is IQtree. The next worst is GTM, and RAxML is the best. Not surprising that RAxML is the best. It's supposed to be the best. On RNA-Sim 1000, they're all about the same. COX-1 het, again, FastTree is the worst. Whoops. FastTree is the worst. The others are close. On RNA-Sim 10K, you start to see a weird thing happen. Now, RAxML is the worst. And our method is the most accurate. So what's going on here is probably that RAxML is spending too much time doing a search, scoring each tree. Scoring each tree means finding the best parameters, and so it's not able to search the tree space as well. And with the 50K situation, it's even worse. So we don't see IQtree and we don't see RAxML at all for the 50,000 sequence case. And the reason is that IQtree failed entirely. RAxML did not fail, but its tree had nearly 100% error. So we're seeing limits of the best maximum likelihood codes on very large data sets. And the limitation in runtime was a week. We didn't let them run for more than a week. And yet this pipeline is able to get good results, better than the other methods on very large data sets. And it's interesting because if you look at it, the pipeline is using IQ-tree for the guide tree. It's using IQ-tree on the subsets, but it's more accurate than IQ-tree on the final thing. So again, divide and conquer can improve accuracy in some cases. So what about maximum likelihood scores? So we took a biological data set from Kelly Williams from Sandia Livermore, and this has like 200,000 sequences, and we took the different starting trees, one of them coming from GTM, and just let them continue and looked at the maximum likelihood scores over time, and this is over a week of analysis. And what you can see is that if you start off with GTM, you end up with a better ML score. You just maintain an improved ML score along the way. This is very preliminary data. We haven't actually finished this part of the analysis. So the overall summary is that large-scale phylogenetic tree estimation is very complicated, but it's becoming much more feasible. Large numbers of sequences are no longer the main impediment. Heterogeneity is an impediment, but methods such as Astral are being developed that address the heterogeneity, and in combination with divide and conquer, you can actually allow these methods to scale to very large data sets. What I haven't discussed here is phylogenetic networks, which is when you have evolution that's not tree-like, genome rearrangements. It would fit into this category, but I haven't looked at it. And multiple whole genome alignment, which is part of the input to these multi-locus problems, where it's possible that divide and conquer could also help. So disjoint tree mergers, the summary, they enable scalability to large data sets. They maintain statistical consistency if you design them well. They improve speed and accuracy compared to Astral when you combine them with Astral. There's a potential that we haven't really yet worked out for improving maximum likelihood search. And GTM is the current leading disjoint tree merger, but that's based upon empirical performance, not based upon theory. The fact that it doesn't allow blending has got to be an issue. So it can't be the best we can do. So now I come to my open problems. So from an empirical perspective, it would really be good to do something better than DTM, than improve on the guide tree measure, GTM, not DTM. GTM is great, it's fast, but it doesn't allow any blending. So if that guide tree has some false positive edges, which it will, and if you are dividing based upon that guide tree, you're going to have a limitation in what you can recover. There has got to be a way of improving this by allowing blending that is still scalable and that maintains good accuracy. So this is an empirical problem that I really would love someone to address. So that's one really basic thing, because it's very generic. Once you have one of these techniques, you can apply it all over the place. What about trying to improve supertree methods? I've told you we have given up on supertree methods, but maybe that's premature. Maybe there are ways to advance divide and conquer by improving supertree methods in that context. So not generic conditions, but conditions that arise during divide and conquer. Supertree methods would have an advantage for accuracy. They're not limited by the division into subsets having some problems. So a better supertree method that's scalable would be fantastic. And then thinking about this in the context of phylogenetic networks is also very valuable. Okay, so those are three empirical directions. Now, theory. Are any of the divide and conquer pipelines absolute fast converging? I've shown them to be statistically consistent, but that's easy. Proving absolute fast convergence is not easy. When I showed you that DCM1 pipeline, that was a way of giving a generic pipeline for absolute fast convergence. I don't know if that will work in the context of these disjoint tree merger pipelines, or even a supertree-based pipeline. But can we get any kind of proof of absolute fast convergence for a divide and conquer pipeline? Theory question, okay? Can we bound the error in the output based upon properties in the input? In what, like the guide tree merger, we have a guide tree. Can we say something about the error we'll have in the output based upon the error in the guide tree? Can we bound it? Maybe. And can we develop a theoretical framework for why GTM boosting is improving astral accuracy? I think I have an idea, but I don't have anything really fleshed out. So some empirical stuff and some theoretical stuff. And now I'm just going to acknowledge, the people who were part of this work. Erin Malloy, who developed these disjoint tree mergers. Paul Zaharias, who was a postdoc of mine and did a lot of the development of this work. Minyuk Park, who participated in the development of it. Vlad Smirnov, who came up with the absolutely surprising guide tree merger, and which worked better than we ever expected, and I still don't know why. And then Kelly, who helped us with really testing it in the maximum likelihood case. And then on the left, I have pictures of Mike Waterman, who was my postdoc advisor, and Gene Lawler. Gene Lawler was my PhD advisor. So what Gene, most of you don't know Gene. He was one of the very first people in computer science to work in computational biology. He, along with Zvi Galil, Gene Myers, were among the very first people from the computer science algorithms community to work in computational biology. And he fell in love with these problems, and he's the one who introduced me to phylogeny estimation as a graduate student. And so he had a huge impact, and also he was someone I loved very much. And I have to just acknowledge that his inspiration and his kindness and generosity was a big part of continuing to love research and just staying with loving research, and that's why you do it. Okay, thank you.
Thank you. So Tandy, back over here on your left. Hi, Terry Gasterlin.
Yeah, I could see you.
Well, first, thanks for the words about Gene Myers. He's been an inspiration in many ways to all of us. And I'll remark on him and then ask my question because it segues. Gene turned his love for algorithms and, in effect, overlapping DNA sequences into assembling the human genome with Craig Venter. One of the two. And that was kind of the big problem that he worked on. So my question to you is, as you've created these algorithms, and it really is creating them, they're coming from in here, your head. What drove you? And what were some of the biggest tree building problems that you've been able to attack now because you've created these algorithms?
Okay, so Terry, first of all, thank you very much. Before I answer your question, I just want to say something that I remember hearing from Gene Myers a long time ago as well, which is that when you're developing methods for use as opposed to for theory, there's this constant feedback between you develop something and then you test it and you discover something about it and then you change it and you evaluate it again and you test it. And this feedback between data and algorithmic thinking, the feedback between data and theory was the thing that really drove his method development. And when I heard him talk it that way was before I had actually been doing it that way. So he also was part of my understanding of that. I'll just add that I went to Arizona for 1 year in my life because I wanted to work with him. And before I got there, he said, well, there's a possibility I won't be here. Not sure, but possibly I'm going to leave and work on, something with the genome assembly. And then, of course, he disappeared, and that was the launching of Gene. Okay, so Gene is amazing, and he had an impact on my life as well. Now, back to your question. What drove me? To be honest, what has driven me was how interesting these questions are. And phylogeny estimation is one of the most mathematical aspects in computational biology. It's so mathematical. There's so much you can do about proving theorems, and yet the theoretical perspective is never enough. And the insights you get into the theory by looking at data were so interesting to me. So what has actually driven me is how interesting these questions are. So as a mathematician, understanding the math questions and making them work on data is a tremendous inherent challenge. That's really not about the biology, okay? Now, making things that are relevant to the practitioner is a huge delight. It's also really hard. It takes years before anything you do becomes relevant. As a graduate student, nothing I did, I think, was ever relevant to anything in practice. What I did as a postdoc, nothing. What I did as a faculty member started, but it was a few years before it was really relevant. So I think you just have to be patient about being able to do something that will actually be relevant, but you keep your eye on the prize, which is you do something that's relevant, and it's part of science, not just part of computer science theory, right? So yeah, you have to stay engaged, you have to keep with it, you have to not give up. Your other part of your question is what did I do that actually enabled things? The development of Astral, which came out of our working on the avian project and discovering the problems there, that development also led to things like statistical binning, also again with Siavash-Mirarod, and it led to the discovery that we had to develop something better than what was currently available. Astral was not yet available, and we developed it and then used it in the plant, the 1KP plant phylogeny. So as we discover limits of methods on data, we develop better methods that can be used on data. A similar thing happened with multiple sequence alignment. The best methods that were available, we thought we had solved it, multiple sequence alignment, but in working with Jim Liebensmack on the 1KP, we discovered that the best alignment methods we had, this again is sequence alignment, didn't do well, and we discovered the problem of sequence length heterogeneity. Sequence length heterogeneity meant we had to change the algorithms we were developing. We developed new methods. So to the young investigators out there, when you look at real data, when you look at biological data, you discover things about data that don't work well with your methods. You understand that, and it leads to new questions, and it leads to new algorithms, and over time it leads to things that actually are way better than the current things. So that engagement with data, both simulated and biological, and that attention to accuracy. If you focus on accuracy, you can figure out the runtime later. Focus on accuracy. Try to give a more accurate result. And then you have this transformation. So Terry, I hope that answered your questions enough.
Yeah, it does. That's great. And
Thanks.
you know what this is what you're doing is at the heart of so much that we do. You know, that's heterogeneity. The sequence length heterogeneity is at the heart of, you know, figuring out, does a protein have a domain exclusion, for example?
Yep.
Thanks, Tandy.
It's part of enabling biology, even if you're not thinking directly about the biology. Look at the data. Look at the data. Thanks.
Thank you, Tandy, for your inspiring talk. So I'm going to ask a question now from the online audience, and then we can continue with questions from the room. So the question is from Shengfei Feng. Have you tried AI approach such as graph neural network or over machine learning techniques to learn the tree structure? Okay,
so the question is, have I looked at using like neural networks instead of the techniques I'm doing? So unfortunately, I'm going to give you an answer that, first of all, it's a little bit old, but a few years ago, there was a paper that came out that was looking at how to do phylogeny estimation by using deep neural nets to get quartet trees and then combining the quartet trees. Now, this is actually relevant to divide and conquer. One of the things that we have discovered is that there's this general tendency to do way better if you have the full data set than if you try to get subsets and combine them. In general, analyzing the full data set at once is better, more accurate. It's just a runtime problem, usually just a runtime problem. So there was this paper that was published that showed that you can use deep neural nets to get quartet trees then under complex models, and then you can combine the quartet trees. And their comparison was mostly in comparison to distance-based methods and on small data sets, simulated data sets and biological. And what we showed was that you could replace that entire approach with just a basic like parsimony analysis on the full data set which would still run because they weren't looking at anything big, and get something much, much, much better. You could even do better by doing a distance-based method on the full data set and get something that's better. So the one time that we have looked at this, the use of deep neural nets was not as accurate. And the main reason it was not as accurate is that it was trying to get quartet trees and use those as building blocks to get the full thing. And the quartet trees, each of them estimated independently, is not as accurate as estimating the full data set and then inducing quartet trees. So my answer is that's all we've done in this area so far, but we've shown a limitation to methods that are based upon whatever technique if you're trying to get small trees only and then extend. So that's my answer.
Thank you. So we can take a question there.
Thank you very much for an inspiring talk. My question is probably a bit basic, but so Astral starts with aligning the sequences, right? My question is there methods that use alignment-free methods to compare sequences? And if not, what is the rationale that doesn't allow us to use them?
So Astral is a method for getting species trees, and the input to Astral is the gene trees. If you don't have gene trees and you just have sequences, then, as you say, Astral would first try to get gene trees by getting alignments and then getting the gene trees. So I think what you're basically asking is about alignment-free tree estimation, not necessarily multi-locus ILS-based question, just the question of alignment-free tree estimation. And that's another example where there are some really interesting methods that have been developed, but so far, I'm not sure that I have ever seen any method that has been as accurate as running, for example, fast tree on an alignment that you get on those sequences. So the challenge for me is the accuracy issue and what you're comparing to. There may be some newer methods that have come out that are actually way better, but I haven't seen them. So it's an accuracy issue. Does that answer your question?
Yes, thank you very much.
Sure.
Hello, over here, to your right. So, thank you very much for your talk. That's fascinating. I was wondering, how are the disjoint sets chosen, and how does that decision affect the accuracy of the tree? Thank you.
Okay, so I didn't discuss it, but the question is, in these divide and conquer techniques for disjoint tree mergers, how are we getting the subset trees, right? Okay, so in GTM, the guide tree merger, there's a tree that's estimated, right? And you take that tree, that initial tree, and you pull out edges until you decompose this into small trees. How small is a function of how small you want it to be. So basically, there's usually an upper limit that's allowed for your analysis. Maybe for Astral, it might be 1,000 sequences or 100 sequences, okay? When I say sequences, I just mean individuals. So again, the idea is you take an initial estimated tree and you pull out edges, like edges that split the tree into two parts, and take out another one that splits it into two parts until all the parts are small enough. So it's absolutely based upon getting an initial tree and decomposing that initial tree. You might ask, what if we had done a different kind of decomposition, like a random decomposition? For the guide tree merger, it would be a disaster to do something random, because you really want them to be true subtrees in the final tree, because you're only connecting things with edges. But in related work, we've looked at what happens when you have random decompositions. It's terrible. Well, at least the first iteration is terrible. If you allow multiple iterations, you can recover. But the decomposition does matter. And so we're always using an initial estimate of the tree to decompose into subtrees that should be reasonable. Did I answer your question?
Yes, thank you.
Thanks.
Hi, Tandy. This is Abhiman from Corteva AgriScience. So my question is more practical. So in the methods and approaches you mentioned, does the speed and accuracy change whether you use a DNA alphabet versus an amino acid alphabets?
Okay, so DNA versus proteins. So my preference when it comes to alignment, if you have coding sequences, is to do it based upon the protein. But my preference for phylogeny is always DNA. And the reason I prefer DNA is you have more change. You really want change to get the tree. You don't want change if you want to get the alignment. But the more change you have, in some ways, the better for the tree. So it helps you resolve the short edges. So that's not a runtime statement, that's an accuracy statement. So again, for alignment, if you have coding sequences, protein, fantastic. And then you can back translate to the DNA and do the phylogeny. Now, how much improvement you get in accuracy, I'm not sure. But we're always doing that. In the various phylogenomics projects I've been on, when we've been doing trees, we've actually often been masking the third position in the codons because they're under selection in ways that the other positions are not, and they're misleading. So we are often processing the DNA to mask the sites that are inconsistent with the model. So that's another point of using the DNA. Okay.
Very interesting talk. Fairly far out of my area of expertise, but very thought-provoking. So I'm going to ask a question that's a little bit more off the wall then. Do you think you could use these phylogenetic approaches to do phylogenetic analysis of like cells from single cell for looking at not distance from sequence homology, but rather expression values or something like that, to trace the phylogeny of cells in disease?
I think that's a fascinating question. And since I haven't worked on it, I really don't have a good answer. But what I would say is if I understood what you were suggesting, you seem to be suggesting using expression levels to get the phylogeny. My preference would be to get the phylogeny somehow that's independent of the expression level, and then look at how expression is changing across the phylogeny. But how to get the phylogeny I don't know.
Well, I mean, I think that would be hard. I think using the differences in expression in the different individual cells, you could use that change as a measure of distance between the two different cells.
So I don't know if that would work. I mean, again, I could really be wrong. It's not my area. So, but in general, I would imagine that expression may not correlate well with evolutionary distances, that you can have expression going up or going down in a short period of time, and therefore probably not relevant to phylogeny estimation. I would try to get the phylogeny first and then look at that. But again, this is not my expertise at all.
Thank you.
Yeah.
Thanks, Tandy. That was awesome. I stole a slide of yours at some presentation before, the one that goes up to 3 million years, where you talk about, evolution at 3 million years.
It's a cartoon. Please don't take those numbers seriously.
Stole it as a cartoon, right?
Okay, good.
The whole point was to say that we can go up to 3 million years back. But I'm interested in 4.2 billion years back, right? So how far back can we go in sequence? At what point do we need structure? And what can you tell me about going back as far as possible?
So I think, so the question is, how far can you go back using just sequence-based analysis? I mean, that's the basic question. So you have an alignment, you try to get a tree. Well, first of all, if you can get an alignment, that's great. In some cases, you have a hard time getting an alignment. The genome rearrangement approaches, we're actually trying to get at those deep evolutionary events that happen rare enough. So duplications, inversions, transpositions, fissions, fusions. My personal feeling is that those techniques are going to give us a signal that goes really deep, and that you again could combine that source of information with sequence-based. You're asking about structure. The ability to accurately infer structure really gives you an additional source of information that's really helpful. I am not discouraged about sequence-based approaches yet. I think a lot of people are discouraged about them because of alignment. They think they can't get alignments. That's why I also work on alignments. So I think the answer is, I hope that we don't have to give up one and use only the other, and we should be using both. But I also don't know what the real limits are to how deep we can go with purely sequence-based approaches.
So can you convert structural alignments into sequence alignments that are meaningful to evolution?
Converting structural alignments into sequence alignments. To some extent you can, of course, but it's the interpolation of in between the established structures that becomes challenging, right? You have this structure, you have this structure, but in between you don't know the structure, right? And so when you're trying to do like a pairwise alignment or a multiple sequence alignment, figuring out how to interpolate between the things that you are sure of, I think that's the challenge. But maybe those can be done with regular alignment methods.
I think I need to talk to you about this.
Oh, we'll talk about it offline? Okay, good.
Thank you. And one last quick question from the online audience. So it's from Hesam Motazeri, asking, so he's saying, great talk. How easy would it be to extend your approach to phylogenetic networks?
So can we extend the divide and conquer techniques to phylogenetic networks? There are divide and conquer techniques that have been developed for phylogenetic networks, and I think that they haven't yet succeeded in really giving the results we need, but they're in the right direction. The big challenge for phylogenetic networks is that when you construct them, when you combine two networks on overlapping sets, you have to be able to reconcile them not as a tree, but with additional edges connecting them. And there's always multiple ways to do that. The big challenge in phylogenetic networks is that you can get a better and better fit for your data, but with added complexity. Avoiding unnecessary complexity is the big challenge. And I think when you're doing a super network approach, you have to figure out how to do that. How to combine two networks in a way that works for your data and doesn't add unnecessary extra reticulations. So the answer is there's a direction to go in there, but there has to be this consideration for the avoiding too much added complexity. So, was that my last question? Okay, thank you all.